Welcome to Click for Biology. This is video number 3 in our topic, which is diversity of organisms. And this is one of 2 videos in which we'll be looking at chromosomes and genes. So videos 3 and 4 are looking at chromosomes and genes. There's a PDF of this presentation that's down in the description below. And down there as well, you'll also find a link to 10 questions. Do those questions to check your understanding of the content of this video and check out the answers to secure your learning of this part of the course. So the diversity of organisms, but we're considering chromosomes and genes. Now, if we look to an overview of the diversity, we're aware, of course, that for sexually reproducing organisms, that their gametes are often in the haploid state with so-called N, designated N number of chromosomes. The diploid state would be 2N. But moving beyond the basics, what we start to discover, and particularly when it comes to species in plants, that there may be what is known as polyploidy, in which we get multiple sets of chromosomes. So rather than the single set of chromosomes in haploid, the double set of chromosomes in diploid, we can end up with 3, 4, and 6 sets of chromosomes, all emerging from speciation events, creating reproductively isolated populations. So we'll take a look at this one as well. On the other hand, it's often thought that the diploid number is unique to each species, and whilst every member of a species, so for instance Homo sapiens, each member of the population of human beings, each individual will have 46 chromosomes as their diploid number. The 46 is not unique to human beings. That is also the case in some of the carp species and in fact in the deer mouse. So the number 46 is not unique to human beings. That is, the number isn't, but there are other aspects to that number. Now, for instance, whilst we've just identified here in the chromosomes that we look at in a species that the number allows us to some extent to identify what we're dealing with, we should also look to the size of those chromosomes and also the position of the centromeres. And these 3 factors will allow us to distinguish, for instance, if we had a cell with human 46 and a carp 46, whilst they may have the same number of chromosomes, they would not have the same number of genes. They would not be the same size, and their centromere positions would be very, very difficult from each other. So we can identify things more carefully through the structure of the chromosomes beyond the number. One of the emerging features that we'll say here and repeat later on is that it's a mistake to believe that the number of chromosomes which is present in some way correlates with the complexity. That is not the case. And as if to illustrate that, we find that the diploid number for the domestic dog is 78, and indeed it is for the turkey, unless of course they might in some criteria be more complex than ourselves. And of course that in itself is an issue about defining complexity. So we mentioned that the chromosome number and the chromosome, the number of genes, the size of the chromosomes and the positions of the centromeres. And when we look at those, what we discover are 2 broad patterns, and that is that individuals within a population, within a species, their variation, since this is the topic that we're looking at, so the diversity within a species, intraspecific diversity, is a feature of different allele frequencies. So individuals within a population will have different alleles for a gene, whereas different genes would be an aspect of interspecific diversity on top of chromosome number, chromosome size, and centromere position, but we would actually also find different genes in those. There are some, through more contemporary analysis of chromosomes in different species, we reveal some very interesting phenomena, as seen here in the analysis of the chimpanzee and the human chromosomes. Now, chimpanzees, and in fact all the apes, have 48 as their diploid number, whereas human beings have 46. If we compare chromosome 12 and chromosome 13 to chromosome 2, 12 and 13 are relatively normal sized chromosomes, but chromosome 2 in human beings is the largest chromosome which is found in the human karyotype. Whilst the chimpanzee and the human beings are well established as in a speciation event and have reproductive isolation from each other, we find evidence of that isolation in what is called chromosomal fusion, in which chromosome 12 and chromosome 13 in the last common ancestor would appear to have joined together to form chromosome 2. This appears to be a malfunctioning of what is known as the DNA repairing mechanism. The process is known as telomeric fusion. Let's take a look at this in more detail. Here I have two different images. One is diagrammatic on the right-hand side, and on the left-hand side we have photographic image. So if I could explain this, these two here and here, these are both the chimp homologous chromosomes, pair 13. This one and this one, this is the homologous pair for chimp homologous pair 12. But this chromosome here is human chromosome 2. And you can see, as I mentioned before, that the human chromosome 2 is very, very large. So why do we think that this has been formed as a consequence of the fusion of chromosomes 12 and 13 in the last common ancestor of us and the chimpanzee group? When we look at the detail, the evidence for this comes from the presence of what are called vestigial telomeres in the middle of chromosome 2, which is in this position here. Now, vestigial means a structure which was present in an ancestral population, but which is no longer functional in the descendants. So this vestigial telomere on human chromosome 2 is no longer functional as a telomere. But we can trace that back to the ends of chromosome 12 and chromosomes 13. There are also vestigial centromeres in both the long and the short arms of chromosome 2 for human beings, and those are in these locations here and here. And those, of course, are inherited from chromosome 12 and from chromosome 13. And once more, these are described as vestigial centromeres, as they no longer function as the human chromosome 2 centromere. It has its own centromere for use in human cell division. And so we have these ancient artifacts, vestigial structures, which reflect the fusion process, the telomeric fusion process, which occurred in between chromosomes 12 and 13. We can go deeper by using a staining pattern called G-banding patterns, and we find that the patterns which are found, you can see here, these dark banding patterns, like so, correspond to the banding patterns which are found when we stain human chromosomes and chimpanzee chromosomes. And so we find that we have evidence from the banding patterns there. We also have evidence from the order of the genes. And so if we come back across here to chromosome 13, the order of the genes in this section is homologous to the order of the genes in this lower section of the human chromosome, and that correspondingly happens in the upper part of human chromosome 2, where these chimpanzee genes are homologous to the upper genes in human chromosome 2. And so the order of the genes which is known as Synteny, is present as well. So that's our fourth piece of evidence. And then finally, we have to note that all of the great apes have 48 as their diploid number, but human beings have 46, showing that 2 of these chromosomes, 12 and 13, have become reduced to form chromosome 2. So there's some evidence of how the change in the diversity of the great ape group may have come about through chromosomal events. It's unlikely that those events entirely account for the speciation of human beings from chimpanzees, but they will reflect the reproductive isolation that they have achieved. The next video goes on to look at chromosomes and the genomic size, diversity in all of the species and all of the living things. There's a chance in the center there to subscribe to the channel. That would be very much appreciated. And of course, the playlist is on the right-hand side for your quick reference. Thank you for listening.